one gene, one enzyme hypothesis. Gene actually do two main functions. One, replication or self reproduction, and second, phenogenesis. Phenogenesis means mechanism in which phenotype of an organism is produced under the control of gene, that is phenogenesis. So we know genes are responsible for development of characters. Also it was called as a trait. So we can say and we will see here how genes they express and what is the relation, particularly relation of genes and enzymes. So concept, that is one gene, one enzyme hypothesis. So genes actually they are hereditary units, also called as factors by Mendel, and they are arranged in linear fashion on the chromosome. All enzymes are proteins, that we know. All enzymes are proteins, and enzymes are also called as functional proteins. Enzymes are necessary for biochemical reactions in the cells. Synthesis of enzymes is regulated by the genes, and how it happens, that we are going to study it here. All the genes present in the nucleus are not active at one time. Remember this thing, all the genes they do not remain active at one and the same time. And here particularly in nucleus, whatever genes are there, out of that all are not remain active at one time. When genes are active, they produce enzymes which affect certain character or phenotype, or also we can call it as a trait, one and the same. It means when gene is active, the enzymes are produced, and these enzymes affect the phenotype. Therefore, according to the physiological need of the cell, synthesis of enzyme is regulated by the gene. Means whenever required, enzyme will be produced, particular type of protein will be produced, and that is responsible for the development of the character or phenotype or trait. Specific genes control the formation of specific enzymes, and this concept is called as a one gene, one enzyme hypothesis. So one gene, one enzyme hypothesis was proposed for the first time by Biddle and Tatum, scientists named as Biddle, B-A-D-L-E, and Tatum, T-A-T-U-M. In 1940, they put forth this concept of one gene, one enzyme hypothesis. They suggested that genes direct the synthesis of enzymes in the cells, which is known as one gene, one enzyme hypothesis now. According to this hypothesis, every gene is supposed to be responsible for the formation of its own specific enzyme. In short, one gene synthesizes one particular enzyme. Biddle and Tatum, they worked on fungus, and name of that fungus is Neurospora crassa. Neuro, Neurospora crassa. And they propose that biosynthetic reaction takes place in a stepwise manner. Each step in the reaction is catalyzed by specific enzyme. So concept of Biddle and Tatum is the reactions occurring to demonstrate one gene, one enzyme hypothesis while working on Biddle and Tatum. So they are of the opinion that these reactions takes place by biosynthetic reactions that takes place in a stepwise fashion, and each step require one specific enzyme. So this thing we can see here with the help of the diagrammatic representation. So here you can see substance A. So this substance A is converted into B, and while this conversion, let us take it as a first step, conversion of A to B, then conversion of B to C is the second step, and conversion of C to D is the third step. So in stepwise manner, the biosynthetic reactions takes place, and each reaction, so this is the first step, second step, and this is third step. So each step require one specific enzyme, and this enzyme is produced under control of particular gene. So only when particular enzyme will be synthesized if that particular gene is expressed. So see here, this is gene 1. So this gene 1, it becomes functional, or it becomes active, and it produces an enzyme, and that enzyme is named as a E1. So substance A will be converted into B when G1 gene produces E1 enzyme, and in presence of this enzyme, conversion of A to B is possible. This is the first step. Now, what will happen if there is mutation or sudden change in the G1 gene? Sudden change in the G1 gene, we call it as a mutation. So if there is sudden change in G1 gene, then E1 enzyme will not be produced. And if E1 enzyme is not there, conversion of substance A into substance B will not occur. You might have cleared the thing. So if there is mutation in the G1 gene, E1 enzyme will not be produced, and therefore in first step the substance A will not be converted into substance B. Now what will happen then? As the reactions are stepwise, so unless until B is produced, second step will not proceed. So B is precursor of what? Second reaction. So sorry, A is precursor of second reaction. Actually B should be produced, and B will be produced only when G1 gene expresses and forms even enzyme. So if there is mutation or some change in the G1 gene, then enzyme E1 will not be synthesized, and hence substance A will not be converted into substance B. As a result, the end product, see here what is the end product of third reaction, third step, that is D. So end product D will not be produced. If we supply substance B, suppose it is mutated, G1 gene is mutated. In such case, if we supply substance B externally here to this second step, externally substance B is provided to the second step, then one can see that reaction proceeds normally and product C will be produced, conversion of this B into C, and C will be converted into D. So all three stepwise reactions or remaining two stepwise reaction will occur if we provide externally substance B to the reaction. Biddle and Tatum proved that each chemical step of a reaction in a cell require specific enzyme that is produced due to specific gene. Means just see here now, if there is mutation in the G1 gene, then B will not be produced. But if there is mutation in the G2 and G1 is normal one, so G1 will produce E1 enzyme. Conversion of A into B will occur. B is reactant of the second step. And now if there is mutation in the G2 gene, E2 enzyme will not be produced. And in absence of E2 enzyme, conversion of B to C will not occur. As there is no C, the D will not form. So like this, these two reactions, these stepwise reactions gets affected if the G2 gene gets mutated. Third possibility, if there is mutation in the G3 gene, there will not be harm to first two reactions, that is conversion of A to B will occur, B to C will occur, because gene G1 has produced E1 enzyme, that's why B is produced. Then conversion of B to C because of the functional nature of G2 in a normal manner to produce E2 enzyme. So there is formation of C. But if mutation is there in the G3 gene, then conversion of C to D will not occur. So C will remain like that. And what our expectation, end product will not occur or will not be produced. That is there. So this is how the stepwise reactions gets affected whenever there is change in the gene structure. And based on this concept, Biddle and Tatum proposed one gene, one enzyme hypothesis. And I have told you that while working on, or they take a tool, experimental tool as a Neurospora crassa for their this experiment. Neurospora crassa normally it is a fungus, and normally it can convert glutamic acid into end product arginine. See here, in a sequence stepwise manner, there is glutamic acid here. So if glutamic acid is produced, ornithine will be, if glutamic acid is there and if even enzyme is produced, the formation of ornithine will occur, and even enzyme production is because of gene G1. So ornithine will be converted into citrulline when if G2 gene forms E2 enzyme. So this E2 enzyme is responsible for conversion of substrate ornithine into citrulline, and then citrulline will be converted into arginine after expression of or formation of E3 enzyme by G3 gene. If these three genes they behave normally, and if they produce their specific enzymes, the conversion of glutamic acid to ornithine, ornithine to citrulline, and citrulline to arginine will occur. So here Biddle and Tatum, what they did, they exposed conidia of Neurospora crassa. They took conidia and they exposed this conidia to X-rays. So what X-ray will do? They found that after exposure of the conidia to the X-rays, they found that fungus was unable to convert glutamic acid to last product arginine. And why this might have happened, they think over that, and they thought that X-ray are responsible for making some change or drastic change in some of the gene, maybe G1 or maybe G2 or maybe G3. So this was their conclusion, that due to X-ray exposure, the conidia, the genes in the conidia, they gets mutated, and that's why their ability to convert glutamic acid into arginine was lost. X-ray treatment induced mutation in gene which could not produce its specific enzyme. So the reaction was blocked by supplying intermediate products. If suppose there is a if we supply B, means, or one can take the example of these products C1. If we try for supplying, if we supply ornithine, and if last step, that is formation of arginine occur, then one can predict that there was mutation or there is mutation in G1 gene. And even though if we provide ornithine to the reaction sequence, then also there is no formation of arginine, then try for supplying the another substance, that is citrulline. And if after providing citrulline there is formation of arginine, then one can surely say that there was no citrulline, means the gene G2 was mutated. That's why no formation of E2 enzyme. And like this, we can have idea that after exposure of conidia to X-rays, which gene might have mutated. By supplying intermediate products, Biddle and Tatum proved that for each reaction, specific enzyme was necessary that was produced due to specific gene. So this is what we have seen related with the one gene, one enzyme hypothesis. So what should be remembered? See, in sequence wise, these stepwise reactions, glutamic acid forms ornithine, then ornithine forms citrulline, and citrulline forms arginine, like this. So a very important concept given by Biddle and Tatum we have seen in this lecture. And remember, this can be asked for either 5 marks or can be asked as a long answer question. So we will have to remember these substances, and the description accordingly you can do. Okay.